Another common organelle in both plant and animal cells is the ribosome, which is present in all eukaryotic and prokaryotic cells. However, the ribosomes in eukaryotic cells differ from those in prokaryotic cells. Ribosomes are tiny, non-membrane-bound organelles that are synthesized in the nucleolus of the cell's nucleus. They consist of two subunits, a small subunit and a large subunit, both made up of ribosomal RNA and proteins. In eukaryotic cells, the small subunit is typically around 40 S, while the large subunit is around 60 S. In prokaryotic cells, the subunits are smaller, measuring 30 S and 50 S. Unlike many organelles, ribosomes lack a membrane, which makes them unique. They are very small, typically about 20 to 30 nanometer in diameter, and appear as tiny, round structures under a microscope. Ribosomes can be found either free in the cytoplasm or attached to the rough endoplasmic reticulum, contributing to the formation of the rough endoplasmic reticulum. Additionally, ribosomes are present in mitochondria and chloroplasts, in plant cells, where they carry out protein synthesis specific to those organelles. These ribosomes play a vital role in protein synthesis. They interpret the mRNA, which is transcribed from DNA in the nucleus, and use this information to connect amino acids and form proteins in the cytoplasm. Ribosomes are the sites where amino acids are assembled into proteins based on the genetic instructions encoded in the DNA. After DNA in the nucleus is transcribed into messenger RNA, also called mRNA, the mRNA exits the nucleus and enters the cytoplasm. Once in the cytoplasm, the mRNA binds to ribosomes, providing the genetic code necessary to link amino acids in a specific sequence to create proteins. Ribosomes read the mRNA sequence and assist in linking amino acids to form polypeptide chains, which then fold into functional proteins. Ribosomes can be found free in the cytoplasm or attached to the rough endoplasmic reticulum, with protein synthesis being their primary function in both locations. Talking about the protein synthesis, let's briefly cover protein synthesis now, and we'll examine it in detail later. Protein synthesis begins with DNA in the nucleus acting as a template to dictate the sequence of amino acids. The mRNA, processed in the nucleus, is released into the cytoplasm, where it binds to ribosomes. tRNA molecules then carry specific amino acids to the mRNA, and the anticodon on the tRNA pairs with the codon on the mRNA through complementary base pairing. The amino acids are linked together as the peptide chain is transferred from one tRNA to another. After this, the tRNA departs, and the protein undergoes modification. Throughout this process, the DNA sequence ensures the correct order of amino acids, ultimately leading to the formation of a functional protein.